everybody. Nice to meet you all. My name is Lucy Kim, and I'm from Oakton High School in Vienna, Virginia. Today, I'll be presenting about the mechanisms of PSGL-1 restriction on HIV-1, SARS-CoV-2, and other pseudovirus attachment and infectivity. Before we begin, here is some introduction about myself. I am a rising senior, and at school, I'm a part of varsity dance team and some other clubs like Operation Smile, HOSA, and Multi-Education Club. Outside of school, I'm very passionate about the medical field. I am a founder and president of Global Medicine Youth Ambassadors of America. It is a youth organization that focuses on gathering future leaders interested in global health, dedicated to spreading knowledge and awareness about the industry through roundtables. So please join if you're interested. Email me, give us a follow, or even just DM us on Instagram. Our username is GMYA_. Other than that, I was also a student speaker for a Global Health Leaders Conference last year, where I spoke about data gaps and social determinants within infectious diseases. I'm also an alumni of Johns Hopkins Medical Intensive Summer School, and under Thomas Nelson Community College, I am a Virginia Earth Science System scholar that gave me an opportunity to further my knowledge as a NASA Summer Academy research intern at Old Dominion University. And lastly, I am a biomedical and microbiology summer research intern at George Mason University, where I will be researching more about this topic this summer. My current career plans include biomedical engineering to study more about health inequities in college, and lastly, to volunteer more frequently for people who are in need of medical care. So let's get into it. First, I would like to go over some context about PSGL-1. The two questions I will cover are the definition of PSGL-1 and how does PSGL-1 connect to what we already know, especially in the field of global health. So according to various studies done in this field, PSGL-1 is short for P-selecting glycoprotein ligand 1, also known as SEL-PLG or CD162. P-selecting glycoprotein ligand 1 is simply a cell surface glycoprotein that binds to various selectants. Its purpose is to mediate the tethering and rolling of immune cells on the surface of the endothelium for cell migration into inflamed tissues, as visually shown on the picture and the image on the right. Under today's context, PSGL-1 is a host protein recently identified to possess broad-spectrum antiviral activity. Now we're going to dive deeper and explore on how this topic connects and relates to the field of global health today. The mechanisms of PSGL-1 inhibits the infectivity of many viruses existing in our world, such as influenza A virus or murine leukemia virus that prove the broad-spectrum antiviral host factor PSGL-1. It inhibits the incorporation of SARS-CoV and SARS-CoV-2 spike glycoproteins into pseudovirions and impairs pseudovirus attachment and infectivity, which suggests potential solutions and ideas that may inhibit coronavirus replication in PSGL-1 cells, which is critical to the global health industry, especially with the pandemic. This topic is very important in the public health industry, as P-selecting glycoprotein ligand 1 is an interferon that restricts HIV-1 infectivity and has recently been found to possess broad-spectrum antiviral activities like what I've just mentioned. It has recently been found that SARS-CoV and SARS-CoV-2 spike glycoproteins can be impaired through PSGL-1 regarding pseudovirus attachment and infection of target cells, which gives current and future opportunities in the global health industry, especially in biomedical engineering and infectious diseases. In the context of viral infection, PSGL-1 has been identified as an interferon-regulated inhibitory factor involved in blocking the infectivity of HIV-1 and other viruses. PSGL-1 may potentially inhibit coronavirus replication in PSGL-1 cells. Understanding virus-to-host interaction is critical to developing both therapeutics and vaccines aimed at containing the pandemic. Now we're going to move on to the presentation body portion of the presentation, and I wanted to discuss more in depth regarding the function of antiviral activity of PSGL-1 when it comes to immunity, especially regarding the different entries of viruses. So how do PSGL-1 block virus attachment and infection of target cells? In order to understand its impact on SARS-CoV-2, it is critical to go over what we have learned from the past from HIV. Some mechanisms of PSGL-1 researchers and scientists learn from HIV is that it does its job by restricting actin dynamics and sequestering HIV envelope proteins. Through this, the steric hindrance particle of attachment to target cells is understandable and available, which allows PSGL-1 to potentially inhibit coronavirus replication in PSGL-1 cells. More specifically, into SARS-CoV-2 entry, it is critical to go over two parts of the entry. First, the coronavirus spike glycoproteins, and second, the significance of host innate immunity. The coronavirus spike glycoproteins is important in the entry of SARS-CoV-2 entry under the presence of PSGL-1, as it gives the ability of binding to the cell surface receptor on target cells, as well as mediating the fusion between the viral and cellular membranes. This brings us to the next point, which is the significance of host innate immunity. Under the host innate immunity, a larger antiviral state is available through the virus infections, triggering an innate immune response and state. When it comes to analyzing virulent incorporation of SARS-CoV-2 proteins in the presence of PSGL-1, I thought the study from the National Center for Biotechnology Information would explain this topic very well. Each data and image organized chronologically by alphabets from A to E represents different interactions regarding how the protein does its job of causing an antiviral activity under the presence and mechanisms of PSGL-1 restriction, showing how PSGL-1 inhibits the binding and entry of virus particles bearing SARS-CoV and SARS-CoV-2 S proteins. Under the incorporation of S proteins, from the quantification and normalization to viral P24 and the co-transfection with vectors, the PSGL-1 blocks virus attachment and inactivates the infectivity of a hybrid reporter viral particle. This is a model for antiviral activity of PSGL-1 against SARS-CoV and SARS-CoV-S proteins, also again adopted from National Center for Biotechnology Information. It shows how under the presence of PSGL-1, the coronavirus spike glycoproteins and the host innate immunity interact with one another to set up an antiviral state. Here is my conclusions with some key takeaways. The first key takeaway is that PSGL-1 imprinted non-infectious viral particles could serve as a live attenuated vaccine for SARS-CoV-2 infection. The second key takeaway is that S protein of either SARS-CoV or SARS-CoV-2 is a phenotype shared among several other viruses such as HIV-1, murine leukemia virus, and influenza virus. Here are some of the future research plans I've found. The first one is that it still remains unknown whether coronaviruses possess a mechanism for antagonizing PSGL-1. The second key takeaway is that it still remains to be determined whether PSGL-1 is expressed in SARS coronavirus target cells in the lungs, and if so, whether its expression can impair the viral infection. Revealing the inhibitors of SARS-CoV-2 entry and the glycoprotein biosynthesis are extremely helpful and important to recognize, especially with the global pandemic and COVID-19 vaccines. Recognizing the effects and mechanisms of PSGL-1 will signify a lot in the future regarding virus infectivity and societal immunity that will further the society's understanding towards recognizing virus to host interactions in the public health industry, and also help elucidate and confirm mechanisms that cellular host factors regulate viral infection and pathogenesis. This is my references. And lastly, special thanks and acknowledgements to first the Global Health Leaders Conference at Johns Hopkins University staff for giving me another great, amazing opportunity this summer to present my passion, and Dr. Wu for giving me an opportunity this summer to study more and further my knowledge about this topic at his lab this summer, and my family members for giving me unconditional support, and lastly, the audience for listening to my presentation. If you have any questions, please feel free to email me. Thank you.